ahead and I'm going to get us started here. So welcome to our live event. The future of healthcare, is data-driven, preventative, and personalized. And I am so happy to have this gentleman here with me today, Dr. Momo Vuyisich. He is a founding member of Viome and our Chief Science Officer and Head of Clinical Research. And that is a mouthful. You're responsible for so much here at Viome. Momo, welcome. I'm so happy that you're giving this talk today to the Viome audience because it's so important to understand where we are now with healthcare and where we could potentially be in the future so easily. So this is an exciting talk. So I really would love to introduce you. I already introduced you, but say hi to our audience, and then we're going to get started in this awesome talk today.
Great. Let's get started. Yeah, my name is Momo. I'm a biochemist and microbiologist by training, and have been working for most of my career to develop technologies that can translate scientific discoveries into something that can enable people to live better lives. I was not going to just spend my life publishing papers for my own career. So let's get started, Lisa. If you don't mind, start the slides and let's roll. Okay, so this is one of the takeaways from this talk, which is that for chronic diseases and cancers, we're in the dark ages. And I'm showing a picture here of a mass grave from the 14th century when a third of the population of Europe died of bubonic plague. And at that time, we had no science or knowledge or technology to actually understand what was causing it. And so there was just massive number of deaths. And it was completely a mystery as to what was going on. And today, we are unfortunately in a very similar boat when it comes to chronic diseases and cancers, is that when someone gets a chronic disease, we say, oh, whoops, sorry, bad luck, right? Well, it's not bad luck. There's some biology and chemistry happening that leads to that disease. It's just that the society has not invested in using the science and technology that we have to understand the root causes. We have spent trillions of dollars on finding pharmaceuticals that can manage these diseases and to some degree make them more bearable, but they're not cures. And so that's really the frustration that has driven me over the last, almost 15 years to really push the science and technology to the edge and try to apply it to solve this problem. So next slide, please. And so I'm making an argument that we currently, today, right now, possess all the science and technology to prevent all future pandemics, to prevent and cure most chronic diseases, to prevent and cure most cancers, and to slow down and reverse aging. We don't need to wait for any technology or miracle to happen. We have it today. All we have to do is apply it the right way. And academic research that's publishing papers is not the solution. And pharmaceutical research to develop drugs that manage an existing disease is also not the solution, but unfortunately, those two are the two major systems today. So that's why we created Viome, to basically create a new way to identify root causes of chronic diseases and then prevent them. And we obviously want to focus on nutritional strategies to prevent them instead of pharmaceutical strategies. So that's what we are about, but I encourage everyone else in the world to shift to this. And I put together a team of people and wrote an article about how we can do this together, but someone has to do it. Let's go. Next. Okay, so probably since you're all Viome associates, you understand the importance of the human microbiome, but I'm just going to summarize it here that inside and on our bodies, we have about 40 trillion microbes. Those microbes code for way more genes than human genes. So we thought that we were human and we focused on the human genetics and human genome, but it turns out that we're mostly not human. We are mostly microbes, and that if we don't include those microbes in the study of chronic diseases and cancers, we're never going to solve the problems. So that's why we are heavily focused at Viome on microbiome, but we are not only focused on microbiome. We're also focused on the immune system and nutrition and human genes and the whole system, the whole body as a whole, as an ecosystem. So we now know that the microbiome is associated with most chronic diseases and cancers, and that in combination with diet, microbiome is actually more impactful on health than human genome. And this is true for essentially every chronic disease. And this has been now published. This is not something that I have to promote with waving hands. It's actually been published. Next slide, please. So I'm just giving you here a couple of examples of the power of the human microbiome, even though this is obviously a vast topic and I could discuss it for days, but here's just some really compelling examples. So in this particular peer-reviewed article, we have a person who suffered from bipolar disorder, and every psychiatrist and psychologist said that something was wrong with her brain, and they were trying to give her pharmaceuticals that modified the brain chemistry, and none of that worked. And so she was very sick, she was not functional, she didn't have a social life, and so on. So out of desperation, she heard about fecal microbiota transplant. She had a doctor oversee the transplant from her husband to her. And after 6 months, she was completely cured of the bipolar disorder. And then this doctor, 3 years later, after follow-up, wrote this article stating that she was essentially cured and she's completely a functional person. So she has the same genetics, the same diet, the same environment. Her lifestyle is the same. The only thing that changed was the gut microbiome, and that cured her. And that's, to me, a very, very fundamental proof that microbiome plays such an important role in a disease where for the last many decades, everyone has been taught that if you have a bipolar disorder, something is wrong with your brain, it's genetic, and we have to pour pharmaceuticals into you. And so we're breaking those myths big time. Next slide, please. If you go read the literature now, microbiome has been so heavily associated with cancer. Every single solid tumor that has been studied has microbes inside of it, and those microbes are likely playing a very important role in the onset and progression of cancer, and they're playing a role in resistance to immune checkpoint inhibitors. And not only that, but the gut microbiome, which is training the immune system, also plays a very important role in cancer treatment. So what I'm giving you here is a summary of two very important papers. One was published by the NIH, and one was published by the MD Anderson Cancer Center in Houston, Texas. And they did a very simple experiment. They applied immunotherapy to a group of people with melanoma, and they observed that some people responded to that treatment, and they became basically clear of cancer, but some people did not respond to the treatment and their cancer progressed. So all they did is they took poop from responders and gave it to the non-responders. And by doing that, they were able to now convert non-responders into a responder. So the same treatment, the same cancer that has now progressed, the same genetics, the same environment, the same nutrition, everything is the same. The only thing they did was change the stool of a person from whatever they had before to something that they received from a person who responded to immunotherapy, and boom, they became a responder. To me, that's, again, a very causative proof that this actually works, that the gut microbiome plays a central role in the cancer response. And we now know also that even before cancer onset and before cancer therapy, gut microbiome plays a role in the onset of cancer, and that it can be either pro-cancer or anti-cancer. And all of those things we want to figure out so that we can make everyone's gut microbiome anti-cancer so that people don't get cancer. That's the best way to approach this problem. Next slide, please. So to solve these problems, we need to understand the human body at a very deep level and understand it as an ecosystem, as we discussed earlier. So we need to digitize the human body and include the microbiome. And I wrote in parentheses here, don't focus on AI. There's just so much hype about AI today, and AI has its purpose and it needs to be highlighted. AI can interpret data today that we simply could not have interpreted 10 years ago because it's a very powerful computational tool. However, the key point there is that you have to have the right data in order for AI to interpret it. If you do not generate large amounts of highly accurate data that are relevant to chronic diseases and cancers, AI cannot just make stuff up. It cannot just imagine things or come up with some crazy stuff. And so it's extremely important to know that data generation that are high quality, very large, and very relevant to the disease states you're studying are the critical component. AI sits on top of that. And yes, it is a very powerful way to interpret the data. So you have to combine the two. You can't just have AI. And so this systems medicine approach to chronic diseases and cancers would include measurements of all the molecules or most of the molecules in our body, which would include DNA, RNA, proteins, metabolites. It should also include lipids and many other proteins, but metabolites includes all those basically. And we should collect it from multiple sample types, not just blood or not just stool, but multiple sample types. We should digitize the human body with hundreds of thousands of data points, then use AI to understand what exactly is the microbiome and nutrition and human genes and human immune system. What are the determinants that lead to the onset of chronic disease and cancers? And then not more importantly, but just as importantly, how do we actually modulate that system with nutrition to prevent chronic disease and cancers? And that's literally why Viome was formed and what we do 24 hours a day, 7 days a week. Next slide, please. I'm just going to touch upon this. This was a very important topic at the World Government Summit where I gave this presentation a couple of months ago in Dubai. But I want you also to be aware that we currently have all science and technology needed to prevent future pandemics, not to manage them, not to watch them and observe them, not to watch all the people die from it and all the economies suffering from it, to literally prevent them at the point at which they develop. And I'm obviously happy to elaborate on that, but that's not really the point of this discussion. But we have all the technology, combining Viome technology with Moderna technology. Just those two, we can literally prevent every single future pandemic. We have all the technology. We just need to have the leadership in place who understands these technologies, understands their power, and knows how to implement them, versus a leadership who is using like frontline nurses and workers to manage an outbreak. That's how we did it in the dark ages. And so the World Health Organization implemented a strategy for COVID pandemic that's essentially not very different from the pandemics of bubonic plague in the 14th century, which for me as a scientist who understands all this is super painful and frustrating, but it is what it is. Next slide, please.
Agreed. Actually, can I butt in here and ask a quick question? Because
Sure.
really, really interests me. So obviously we have all lived through a pandemic now, you know, the last one was about 100 years ago. So, but what would the fundamental changes need to actually be for World Health Organizations and governments if we were to move over to this model of very quickly handling pandemics before they spread?
Yeah, I mean, the first thing we need to do is change the leadership of these organizations to people who are scientists and who understand science. That's the first thing. Someone who is a medical doctor who knows how to manage an already existing disease is not the right person with the right mindset to prevent pandemics. Their mindset is, pandemic is coming, how do I deal with it? Well, COVID showed that that's not a good approach at all. And so we have to have the right leadership, but that's true for everything, for every company, every government. If you don't have understanding, knowledgeable, capable, competent leadership, well, things go bad. So to me, that's the most important step. And then that leadership needs to understand that the technology already exists. They need to just find logistics to implement it. And I have specific proposals on how to do that. That's not rocket science, you know, and so we can do that.
Yeah, it seems like we are taking baby steps, but it is a big mind shift for sure.
Yeah.
Yeah. All right. I will butt out again and I'll let you continue.
Sure. Okay, I just wanted to throw this slide because this is really one of the takeaways and one of the things that we think about every day. You know, healthy people have many wishes. If you're healthy, you're thinking about big dreams and big plans, and you're motivated. You can do a lot of things. When you're in pain 24 hours a day, you don't have dreams about accomplishing things in life, about having a career, having a family, doing things. All you're focused on is, how do I get rid of this thing off my back, right? And so sick people only have one wish, which is get rid of this disease. And that's a frustration that people who are healthy do not understand, but people who have been sick understand. And this was my frustration for about 15 years, that I had a chronic disease, and that all of my daily motivation now that I'm healthy is derived from that frustration for so long. Next slide, please. Okay, so here's my story. So in my 20s, I was diagnosed with an autoimmune disease. Well, no one diagnosed it. They just said I have autoimmune disease, and I could have had maybe arthritis or ankylosing spondylitis and so on. And so all the doctors wanted, they didn't know what was wrong with me. They just labeled it as autoimmune, and they just wanted to pour drugs into me and basically slow it down. But that would have just slowed it down. I would have by now been in a wheelchair, which is obviously unacceptable. And I was, as a scientist, as a curious person, as someone who was able to read every single tiny piece of information in the scientific literature, I was able to test different things, observe different things. And finally, I found a diet that completely got rid of my symptoms. And I mean, completely, I completely cured myself. It took about a year to heal from all the damage that was done to my body. But since then, I've been now completely healthy, no symptoms whatsoever. And so what that means, just at N of 1, just for me, not only am I able to be a productive member of the society and enjoy every day of my life, but I saved hundreds of thousands of therapy costs, because it's about $100,000 per year in therapy, and then saved hundreds of thousands of dollars in lost productivity, prevented daily pain and suffering, and decreased the risk of other chronic disease, because if you have an autoimmune disease, you have a higher risk of many other diseases. And so all of those things with a diet change. And that's something that the current system does not recognize, does not want to recognize, does not know about. It's scared of it. And this is the revolution that we're leading at Viome. And obviously, we need a lot more help from a lot more people and companies, and we just need to change the whole mindset. But look at what's driving this. Companies are making massive amounts of money off treating autoimmune diseases. So they have not created a business model where if they cure you, they monetize. If they cure you, they lose you. I'm not making them any money, and I could have been making them a lot of money. And so we are creative at Viome, where our business model is to keep people healthy, and that's against basically everyone else who has created a business model for keeping people sick. And so we all have to contribute to this every day in every way that we can. Let's revolutionize the way health is approached. We want to keep health, not manage disease once we develop it. Next slide, please.
Great. You know, Momo, I've been always very fascinated with your story. And I wanted to ask you a quick question. How did you stumble upon, or actually meticulously research is probably a better phrase, this solution for your personal health issues with nutrition?
Yeah, I mean, it's a very long story, but I had tried every diet possible on earth. I had, tracked everything. I had tried the, elimination diets and all that stuff. This particular idea came from a paper published by a UC San Diego professor who was studying this in mice. I was actually reading the paper while my wife was driving through Arizona back from our Christmas vacation. I was on my computer reading every single paper that had ever been published on human health and microbiome and nutrition and chronic inflammation. And this one paper, I read it and light came on. I was like, this makes sense. I'm going to try it. I designed my diet around it. And a month later, I knew this was it. And a year later, I was completely healed and cured. So it's a bit of a longer topic, but if people are more curious about it, the molecule that causes this in me is NEU5GC. It's spelled NEU5GC. And I think that this is a super common issue. I think hundreds of millions of people probably have something that I had. It's just that, they just go through the standard process of going to the doctor and getting the drugs, and they don't know what causes it. And the doctor obviously says, bad luck, right? But I wasn't that type of a person. I knew that this was not genetic. I knew that diet was the key. And finally, I found it. But it was a very long and painful process.
Wow.
Next, please.
We're definitely happy that you made that discovery.
Yeah, I know. Very happy myself. Okay, so chronic disease burden. So in the 1990s, we pushed for sequencing of the human genome. The promise was that once we sequence the human genome, we will cure all diseases. That has turned out to be completely false. Chronic diseases are actually in many ways on the rise. I'm just giving an example here that not only is type 2 diabetes increasing in prevalence in the overall population, it's actually increasing in prevalence in younger population. More and more younger people are getting type 2 diabetes than before. So not only did Human Genome Project not solve this problem, it's gotten a lot worse since the Human Genome Project finished. So that original promise that human genetics was going to solve these problems is completely false. And that needs to be simply revealed and publicized, and scientists need to admit that that was a mistake, that we need to shift now our our focus from genetics to epigenetics. But that's a very difficult shift to make. You know, back in 2010, I completely shifted my scientific career. And every single peer that I had, everyone that I knew in life, they said, that's a suicide. Changing your scientific career that fundamentally, that's a suicide. You will never make it. Just go to management because you're going to be done soon. Well, it takes an extra effort to change your scientific career, but we have to do that because it's clear now that genetics is not going to help us. Next, please. So here we have a peer-reviewed publication, and by a scientist stating facts that genetic factors are not the major cause of chronic diseases. So that's one of many papers that has come out recently. If people are more curious about it, to me, the best evidence for chronic diseases and cancers not being genetic is when they study identical twins. So in very large twin studies, it's already been shown that diseases like Crohn's disease, several cancers, type 2 diabetes, they're all lifestyle diseases. They're not genetic diseases because identical twins that have the same DNA, one of them develops it, the other one does not. And so there's now quite a bit of evidence. I don't have to wave my hands and try to convince people like I did 10 years ago. Now the cat's out of the bag. So anyways, next slide, please. So in other words, the takeaway from that slide is when people say it's genetic, no, it isn't. And even if it is partially genetic, it can still be prevented and overcome with a lifestyle and diet change. So even if genetics plays a role, not everyone who has that higher genetic risk is going to develop the disease. So there is lifestyle and diet that controls that switch, and we have to be conscientious of that. Okay, so now that was all sort of an introduction, some thoughts of mine. And now we get into the real meat of this presentation, which is Nutrition 2.0. And so Nutrition 2.0 is something that we invented at Viome. I sort of imagined it back in 2010, about 6 years before we started Viome. And I have a Ph.D. in chemistry, and I have an undergraduate degree in microbiology. I have run many labs in chemistry, biochemistry, microbiology. So this is sort of my comfort zone. And I thought when I started my journey of dieting, I thought to myself, wait a minute, if I go to 3 mathematicians and I ask them, what is 2 + 2? And they all give me the same answer, I'm like, wow, math is a very objective science. I like that. If I go to 3 nutritionists and tell them the exact same story, what my symptoms are, I get 3 different recommendations and they all swear by it. But if you get 3 different recommendations from 3 experts, given the same input, that doesn't make any of them scientists. They're all guessing. And that's really what triggered me to say, well, wait a minute, how can that be? That's not science, that's opinion. And so Nutrition 2.0 is a quantum leap in nutritional science in that it is 100% data-driven and objective, 100%. So I will focus on this chemistry plus math, but chemistry is a very objective science, math is a very objective science. Today, from the moment that a sample arrives at Viome, until it goes through all of the laboratory analysis, all the bioinformatic analysis, and all the AI models in order to compute the diet for a person. There is no human opinion involved in any step of the way. It is 100% machine-driven and data-driven. There are no opinions, and there are no preconceived notions. Our computers do not know anything about ketogenic diet or Mediterranean diet or anything like that. It is brutally, brutally objective, and that's exactly what I wanted. It is also highly personalized. There is a diet for every person. We have currently something like 350,000 customers in 106 countries, and we have 350,000 diets. There are no 2 people who have the same diet because there is no diet. It's just what ingredients each person needs and which ingredients they need to avoid, and that's mapped to their diet. The goal of this Nutrition 2.0 is to prevent and cure chronic disease and cancers and slow down aging. It's not just some sort of eat healthy. It's truly to live a better life. And an extremely important point is this last bullet, which is it continuously improves by self-learning. So if you go today and do a lipid panel test at Quest Labs, that test has been run for about 50 years. And Quest today, after 50 years and running it 1 billion times, cannot give you any more information from that test than it did 50 years ago, because it has not implemented any science whatsoever. And one of the outcomes of that is that 50% of the people with a heart attack in the United States have a normal lipid panel because we haven't learned anything from it. We don't know what it means. So at Viome, from day one, every single customer that adds data to the database, every single clinical research participant that adds data to the database, we use machine learning to learn from it. So when we talk about chemistry, we are not changing the chemistry. We're still measuring the same chemistry that we've been measuring from the start of Viome, but the mathematical equations that are overlaid on top of that chemistry are continuously evolving as we learn. So no matter how good or bad this Nutrition 2.0 is today, it's better tomorrow, and it's better the next day, and a year later it's better, and it's continuously improving in a very objective way. And that's really one of the critical components that we've added to Viome from the start that's going to continuously drive this process to be better and better. And when I say better, higher efficacy in higher percentage of people until we drive it to 100%, and we know every root cause of every disease, and we can modulate it with diet only. That is the goal, obviously. Next slide. So I'm very, very, very proud of this. This has been 13 years of working 7 days a week, 365 days a year, nonstop commitment, and we're moving along. So how do we learn the math? So this talk is not about the chemistry part. The chemistry part, we've published quite a few journal articles on this. It's basically how do we convert stool, blood, and saliva into digital data? How do we measure the chemistry of the microbes, of their genes, of human genes? All of that has been published. It's out there. We can obviously provide those links, but you can just search for that. And so that's the chemistry, and that has not changed, and that will not change for a very long time, because that is the fundamental chemistry of our body that includes the microbes and our immune system and our genes and so on. So let's talk about the math. So we said at the beginning in my introduction that I'm also the head of clinical research programs at Viome, and that's another thing that we've revolutionized to the point where we made a quantum leap in clinical research programs. We have enrolled 16,000 clinical research participants in 6 years at Viome. I don't know the numbers, but I will be surprised if Harvard University as a whole has enrolled more than 16,000 participants in that same time period. So even though we're a small biotech company, we have been able to perform an enormous amount of clinical research with the resources we have because we're super efficient. And so one of the big studies that we've performed in the US and Japan is to determine how does the gut microbiome determine the glycemic response to different foods. And so just a simple observation here is if you mount a continuous glucose monitor on 2 people and feed them the same foods, you can see that they can have completely reverse response to those same foods. The green line here corresponds to a banana fed to 2 people, and you can see in person 1, it spiked their blood sugar very high. In person 2, it did not. And then purple is the sprouted grain bread. You can see that the effect is reverse. If you look up on Google glycemic index of foods, you will see that a banana has 1 glycemic index for everyone, which is the average person, and that wheat bread has an index for everyone, the same, which is the average one. But it turns out none of us are average. All of us have individualized responses. So Viome launched these studies, and we invested an enormous amount of time and money to learn how each person will respond to every food just based on the gut microbiome. So next slide, please. So just a summary, this has been published, so it's out there, but in summary, we enrolled 1,100 participants, half of them in USA, half of them in Japan. We performed the stool test, that's the chemistry, and then we fed them foods for 14 days while monitoring their glucose. And we measured a total of 60,000 meals and snacks consumed by these 1,100 people. And we provided the food for 40,000 of those meals so that we know exactly what went into them. We had truckloads of food delivered from Whole Foods in order to deliver them to the participants here in the US. It was an enormous effort, but we measured the glucose response to 60,000 meals in 1,100 participants, and we built a machine learned model that allows us now to recommend to someone exactly what to eat and how much of that to eat in order to minimize their exposure to high glycemic response in blood. Next slide, please. And so this has been, again, published. We developed a machine learned model. And now every single customer of Viome benefits from this. This is all built into our app. When I say mathematical equations that we apply today to that chemistry for every customer were learned from these clinical research studies. So they're not, we don't have some smart people who invent them. We don't have those mathematicians. This is simply learned on actual data from actual human beings in a very large setting. And that's the best way to do science. Next, please. So now that we spent a few years developing these mathematical models, and then we wanted to apply them to actual people to see how well this works and measure it, because we don't get to measure that necessarily all the time. So what we've done here is the next 5 slides, I'm going to show you some preliminary data on the efficacy of our personalized nutrition on several conditions. And so here we have people with IBS who take our personalized diet. And you can see here that table summarizes the stats, but the graph may be easier to see. Lisa, do you see my cursor or not? Oh, we see yours, I guess. Okay. So on the right side, the graph shows blue bars and yellow bars or orange bars. The blue bars are the initial levels of disease for example, 52 people, N equals 52. And after 212 days of Viome personalized nutrition, you can see they now have symptoms that are in orange level. So they decrease significantly, and the stats are in the table. This has all now been published as a preprint, and it's under review in the American Journal of Lifestyle Medicine. But you can see here that for IBS, people with severe IBS, moderate IBS, and mild IBS, we were able to make not only significant improvements, but mathematically significant improvements. The p-values are all listed in that table. And now next slide, we show basically the same graph for depression. We see improvement in severe depression, moderate depression, and mild depression. Next slide, we show that for anxiety. Same story. Please note that these are preliminary data. These are single-arm interventional trials. We do not have a control population for this. So that's coming up in our randomized controlled trials that we are running right now. We've already recruited half the participants for those 4 randomized controlled trials that we're running. Next slide, please. We also built a state-of-the-art type 2 diabetes risk score, which was based on 55,000 people. And we show here, we now have a control group. And we show here that the blue people who are a control group because they self-reported to have low adherence to the Viome personalized nutrition, they improved their risk score, but not by a lot. And then the green graph, the green people, they self-reported high adherence. They improved their score much more, and the p-value for that is very low. So this is very good evidence that our personalized diet is actually working. This was also in the publication. Next. And then we have another study that we did, interventional study on depression, where we now have a control group. So this is basically Viome customers who self-reported that they either did not consume any supplements or foods that we recommended, so low adherence, and that's called a control group, and customers who we actually provided the personalized supplements, and we assumed they consumed them, which is a fairly safe assumption. And we can see here for depression that we see a very large difference between people called PS, meaning that they consumed personalized supplements, versus the control people who did not. This is moderate to severe depression as measured by a clinically validated score. Now, please note that even though these data look very good, they are preliminary data. The significance mathematically is not great. It's good, but it's not great. And that we're currently running a randomized controlled trial on depression, which will basically provide us the gold standard data for does this work and how well it works. But certainly what's exciting to me is if you look at the color graphs down below, you can see that people who consumed our supplements we did observe much better improvement in clinical scores. And so this is very promising. If this turns out to be mostly or largely a placebo effect, I would say it still doesn't matter because people are getting better. And that's really what we are about. But of course, we want to keep improving that. So next slide, please. Again, I just want to reiterate that genetics is not the end all be all. There are some genetic diseases that are almost certainly going to cause a disease, but the vast majority, including most of the cancers, are not a genetic disease. This has now been published. So it's not something that I have to preach. Next slide. And we have committed a lot of resources to cancer and aging. On the left side, I'm showing you two graphs from a paper we published showing that we've built state-of-the-art aging models based on both human and microbial gene expression. And that's a very novel concept that no one else has done. On the right side, I'm showing you the results of our Cancer Detect test, which is able to detect oral and throat cancers. This is a commercially available test, and we're actually in 2 weeks launching a provider version of it through a dental channel and healthcare provider channels. So we're very proud of that. It received a breakthrough designation by the FDA for accelerated reviews. And so very proud of the data, very proud of what we have been able to do. Next slide. And I think this is the last slide, and it's sort of a teaser, but there is some really useful information here. If you guys are not caught up on this, Moderna is now running trials for anti-cancer vaccines. And this has been shown recently in a Japanese group that they can actually cure cancer in mice using mRNA vaccines like Moderna. So this is now a rapidly evolving sort of concept where the reason people develop cancer is because their immune system failed to identify cancer cells, and that's why cancer arose. This is called immune surveillance. If you guys want to read more about it, cancer immune surveillance is a very natural mechanism that prevents cancer progression, because we get cancer cells in our body to the tune of billions per day, but the immune system is supposed to find them and kill them. So, but when it fails to do that, cancer actually develops into a tumor. And so, but now we have a technology with Moderna and other companies where you can actually train your immune system that, hey, there's cancer in here and here's what it looks like, go kill it. And so that's a very, very, very powerful approach. And we at Viome have patented an approach that's very similar to that, but that would apply to aging, meaning that we should be able to design anti-aging vaccines, and they should work. In theory, this should work well. And so at some point down the road, when we become profitable and big company and have money, then we will make that investment. But for now, we are in a holding pattern. But that's a very exciting future science that soon we should be able to treat cancer better. Eventually we'll prevent it, and then we can focus on aging, healthier aging, slow down aging, and so on. Next slide. Well, this is an opportunity to do question and answers. These are pictures of downtown Seattle with Mount Rainier in the back. This is where we have two offices. We have our laboratories in Bothell, Washington, just north of Seattle, and our corporate offices in Bellevue, Washington, very near Seattle. And on the right side, I'm showing New York City, where our offices are for all the computational resources. So we have bioinformatics, data science, computational systems biology, all of those super smart people who work with supercomputers every day are to interpret the data and come up with the best mathematical equations. And I will take any questions.
We do. We have a lot. So we'll try to get through as many as we can. We have limited time here. But Momo, if you want to open up your Q&A too, if you want to look, I will read a question to you, but you can look as well. So I believe this is Ina. Says, in regards to fecal transfer, bipolar patients, strange that the husband and wife didn't largely share similar microbiome given their intimate relationship. Was it that they lived completely different lifestyles and had completely different diets? That's a very
good question. Yeah, I see it. Good question. Yeah, no, it's extremely rare that people who cohabitate have similar microbiomes. They have very different microbiomes. And the reason is because think of microbiome as sort of an organ that established itself as you grow up as a human, and it established itself due to your environment in which you grew up and your immune system interacting with it. Both of those things shaped it, and once it's shaped, that gut microbiome wants to stay there. They don't want to just be flushed down the toilet. They want to stay there. So they've created sort of a very complex community where they feed each other and they help each other fight off any newcomers. And that's actually a part of the natural mechanism by which our microbiome is helpful to us. It's that it prevents pathogens and other newcomers from coming in. So this is a very common thing that people who cohabitate, they don't have any more similar microbiomes than people who don't cohabitate. Good question, though. Next one.
Yes. So let's read Kenneth's question. Which tests did Momo have done that showed an autoimmune issue, ANA, anti-CCP, rheumatoid factor, and did those test results return to normal after his dietary self-treatment?
So this was a long time ago, and I no longer remember what tests they ran on me. I know my CRP was off the charts, which is why I knew that I had chronic inflammation. I don't remember the rheumatoid panel, if at that time it even existed. And I don't remember what they ran, but I mean, every joint in my body hurt and every soft tissue hurt if I touched anything. So definitely had a lot of pain points.
All right.
And all of that, my CRP has been, below 1 milligram per liter for the last, 20 years. So next.
Yes. So Sue asks, isn't applying nutrition 2.0 considered using AI?
Yeah, let me clarify that. So we use machine learning and artificial intelligence and data science to analyze the data. The output of that analysis is a mathematical model. And you can think of that mathematical model truly as a complex mathematical equation. And so when we apply those equations as part of V 2.0, then it is not AI, it's just a mathematical equation. Now, remember the last bullet of Nutrition 2.0 is that it's self-learning, and that part is definitely AI. So every single day, 24 hours a day, Viome runs supercomputers in the cloud to analyze the data and self-learn to improve those mathematical equations. I hope that's clear.
Excellent. So actually, another related question, actually, both Ed and Danielle asked about our recommendations. So
yes, I see that.
Yeah. Mm-hmm.
Yeah.
So how is your system continually learning? Yeah.
Yeah, so we use AI to learn from new data that we obtain from our customers and from our clinical research participants. We produce new models, which are essentially mathematical equations, and those are integrated into our results and recommendations. So the example I gave in the talk was we performed this super large study in the US and Japan where we learned mathematical equations that can convert someone's gut microbiome composition and functions into what meals they should be consuming and which meals they should be avoiding. And those mathematical equations were plugged into our recommendation engine, and boom, automatically every customer benefits. And right now we've done now that for IBS and depression and anxiety and type 2 diabetes. Like I showed you the data that, this seems to be working, and we've now launched randomized controlled trials. So yes, continuous learning is one of the key aspects of Nutrition 2.0 and the entire Viome platform.
All right. Karina had a pretty scientific question. Can you comment on how you deal with genetic mutations when someone doesn't metallate vitamins because of gene mutations?
Yeah, we currently don't incorporate genetics into our recommendations. So we will get to that at some point in time, but we simply don't do genetic screens at this time. That's coming at some point in time. But right now, as you guys know, we sequence RNA and we only do epigenetic studies, not genetic studies.
All right. Irene asks, any practical tips on how to improve an aging microbiome? I like that question. Thanks, Irene.
I mean, I'm not providing here medical advice, just so we're clear, but there are now very, very solid data in animal models showing that if an older animal takes a fecal microbiota transplant from a younger animal, that all of their metabolic parameters and their aging data look better. So that would be sort of the thing, but FMT is obviously still Pandora's box. There's a significant risk in performing FMT. So any practical tips on how to improve an aging microbiome? So one thing that I do that I would recommend to everyone is continuously rewilding your gut microbiome and oral microbiome. And that there's sort of a series of steps, but the main ones are minimize consumption or avoid antibiotics the best you can. And that includes not just prescription antibiotics, but antibiotics found in foods. Minimize or eliminate consuming preservatives. So when you see a food label and it says they added something to preserve freshness means to kill bacteria, because bacteria cause unfreshness. And so just avoid all those foods, avoid eating out at all costs, and avoid going to parties where there's food served from the outside that you have no idea what's in it, because that's a problem. And then do organic gardening. Don't peel your vegetables. I pull a carrot out of my garden and I just shake off like the major soil, but there's still soil coating my carrot, and I eat it right there in the garden. I do that all the time. I've been doing it for 10 years, and my gut microbiome has increased in diversity massively. So there are a lot of these things. Dr. Robin Chutkan, I think she wrote a book, but she definitely has some information about rewilding your gut. So I would research that. But yeah, anything you can do to be more closer to nature, and rewilding your gut is good. Anything that you do to reduce the diversity of your gut is going to be a problem, because reduced diversity of your gut microbiome has been associated with essentially every disease. That's all I can say for now.
All right. Rick wants to know, how do people get into your studies?
Yeah, so we have on Viome Life Sciences. So please remember that Viome Life Sciences is the company where I work. Viome.com is like an application of Viome Life Sciences. So Viome.com is the nutritional branch of Viome Life Sciences. So if you go to viomelifesciences.com and go to clinical research, you will see the active studies, and you can sign up from them right there.
All right. Excellent. So I'm just filtering through some of these. John asked if this presentation will be available online. Yes, it will. Don't worry. You'll get a follow-up email from us with a recording of this. And let's see. Kenneth asks, is there an underlying ideal microbiome for everyone that we can all target? Is there a perfect fecal transplant that can help achieve it?
Yeah, good question. So people have tried to do that, and they've all measured the composition. There is no composition of microbiome that's perfect for us that we know yet. I think in the future that may be possible. For now, what I would like everyone to focus on is that almost everyone's microbiome can be healthy. And that's because if you feed it the right diet, it will perform the functions that are healthy and not perform functions that are detrimental to your health. So all you have to do is match your microbiome with the right diet, which is exactly what we do, and your microbiome can be healthy. I mean, that is exactly the purpose of our diet recommendations. From our clinical studies, we know what functions of the microbiome are associated with diseases and which ones are associated with health. That's exactly what machine learning models tell us. And then we develop models where we say, okay, how do we prevent the microbiome of a person from performing functions that are detrimental to their health? Well, we deprive them of foods that feed that portion of the microbiome. And how do we increase the healthy functions of the microbiome? We feed them foods that feed the healthy portion of the microbiome for that person. And a different person will have a different microbiome, so we have to provide different diets. That's why everyone gets a different diet, because the formula to feeding the healthy part versus the detrimental part is different. And very often, there are bacteria that have both detrimental functions and healthy functions, and you can feed it just the right material so that it only performs the healthy functions, but not the detrimental functions. And so there's some percentage of Viome customers, and it's a gradient, it's a gray area. So, I don't want to say the percentage, but, single-digit percentage of Viome customers who have such poor diversity and richness of their gut microbiome that they basically have lost functions that are healthy to themselves. They will have to go rewild their gut microbiome in a very significant way, because they don't have the functions. No matter what they eat, they will not get better, because their microbiome cannot convert any food known to man into something beneficial for them. They've basically consumed too many antibiotics, too many chemicals in foods. They've had too many gastrointestinal diseases, and they do not have a healthy microbiome. They cannot have a healthy microbiome. So unfortunately for those people, they'll need to go rewild it. And the probiotics that are available in like consumer products like yogurts, kombucha, and so on, they don't have, or probiotics off the shelf, they're just bifidose and lactose. They're not gonna give them back those essential functions. But for the vast majority of people, they can have a healthy microbiome. They just need to tune it with their diet.
Agreed. So kind of to Off that question, Anna asks, do you have data of studies comparing following only prebiotic and probiotic and dietary recommendations versus diet, pre and probiotics and supplements as well?
No, we do not have that.
Okay, cool. Let's see, one more.
No, but yeah, go ahead.
I'm just reading through all of them here. Oh, Richard asks, what are your thoughts on proteomics?
Proteomics.
Geonomics. Sorry. My understanding is that Viome looks only at the mRNA, but what about proteins?
Hello, Richard. So proteomics is obviously a part of what I outlined up front as part of a systems medicine approach. If we had unlimited amounts of money and our customers could pay unlimited amounts of money, we would certainly include proteomics. What we've done at Viome is, instead of doing a sort of a meta-proteomic analysis of all the samples, we have now developed a multiplexed immunoassay that can quantify proteins that metatranscriptomics cannot. And so we've developed and clinically validated an assay for blood that uses a blood card collection method that is analyzing proteins in blood that are actually not produced in blood, meaning that their transcripts in blood are not going to be informative. So this is, for example, CRP, which is produced in the liver. We can actually quantify it in blood, and then a whole bunch of antibodies that, even though they're produced in blood, we cannot quantify them because we don't know which transcript codes for an antibody against some specific antigen, like a food antigen or a thyroid peroxidase and so on. So proteomics eventually, if they can commoditize, if they can commoditize metaproteomic analyses, we might consider it. But we feel very strongly that for most proteins, especially prokaryotic proteins, the mRNA translates well into protein. Obviously, for human, there are some post-transcriptional modifications and mechanisms that can regulate the protein level different from the mRNA. But those, I think, are edge cases, and we'll address most of those via this Viome multiplexed immunoassay. So all that is in development. We're aware of it, but we have to grow at a certain pace and cannot overstretch ourselves.
All right. So we are at about time. I do have one more kind of a question and a comment from Julie. I wanted to read this one, Julie, because I really liked your question and comment. How have things advanced since the first test that came out? I took the test about 6 years ago. It changed my life. Everything I was eating was everything I should not be eating. I have recommended this test to so many people. I appreciate your work. This field is so important, which is why I am in school to learn more, and will continue to recommend your testing. Excellent.
Yeah, I mean, so our initial platform was based on mostly modulating the human inflammatory responses. And we picked those first because there was a lot of domain knowledge, and we had some preliminary data on those based on our early clinical studies. And those, chronic inflammation is sort of the root cause of many, many diseases. So we did the most impact with the least amount of effort to start with. And then we started adding on. So for example, the large study that I described on the glycemic response, we didn't have that model when we started the company. We had to invest a lot of time and money, and we finished that 2019, I think. So we incorporated those models. So that significantly improved metabolic aspects, not just immune aspects. And then, for example, one of the things we discovered via machine learning was that plant viruses actually played a role in some diseases, and especially IBS. I mean, it's an incredible thing that the world didn't know about that, and we didn't know about that when we started the company, but machines picked it up later. And so we have improved, and we are continuously improving. That's a part of the platform. And we've refined, for example, that initial model of immunomodulation by the microbiome, and we're going to continue to do that. But we did have a very good product to start with that made a huge impact on a lot of people. So, but we're continuously learning.
Absolutely. Onward and upward. All right. So Momo, I want to thank you very much for taking the time to come here today and talk to our Viome audience about this really fantastic subject, future of healthcare. And thank you for giving us your knowledge and your expertise and answering all those questions. And I also want to thank you, our audience out there, for joining us here today and spending this hour with Momo and myself. And I just want to make sure that if you want to continue or start your health journey, I just want to give you a discount code, April webinar. If you use that on our website right now, you can get up to 40% off the original prices of any Viome intelligence test, anyone you choose to do. And please take care. Have a great rest of your day today. And again, thank you, Momo, so much for joining me here today.
Thanks, everyone. Thank you. Good day.
Bye-bye.